I'm Dr. Sue Ishak, and I'm really excited to convene this special session that we have for you this afternoon, titled Microbes and Social Equity, the Microbial Components of Social, Environmental, and Health Justice. So microbial communities are really found everywhere. We, of course, know that. We've been hearing about that for 2 days, and we'll hear about it for 2 more days. So microbes are found in every environment under every context. So there's no microbial ecosystem that does not impact an environment or biology in some way, and therefore impacts a society in some way. So there's really no aspect of our lives which don't involve microbes. And thus we posit that microbiome work has the capacity for social impact and vice versa. So the aspects of our lives that are determined by public policy, for example, how easy it is to access a fresh and healthy diet, access to safe and clean housing and affordable housing, and access to healthcare of any kind, all of these things affect our encounters with microbes. So these resources are known to keep us healthy, but they're also important for helping us have interactions with microbes that might also benefit us and our health. So equality would mean that we all get the same resources, and it, as in the name, literally depends on everything else being equal. And we know that humans and our societies aren't perfect for intentional and unintentional reasons. And so sometimes we just need to reimagine the way that we're distributing things. So social equity is a process of ensuring fair access to resources, and assumes that we aren't all starting from the same starting point advantages. Some people just need a little extra help to reach those same resources, and social equity acknowledged that we don't all need the same type of help. Thus, social equity is really rooted in understanding the life of that individual. And so in talking about microbes and social equity, we posit that social contexts are really required to understanding the interactions between human and microbes in many different settings, whether you're talking about specifically host-associated microbes or whether you're talking about microbes that we encounter in our daily environment. So in order to get the most out of your microbiome research, you also need to gain an understanding of that context, engage in discussion with relevant communities and stakeholders, and then also collaborate with those same groups who are or who have the potential to be impacted by your microbiome work. To that end, I founded the Microbes and Social Equity Working Group back in 2020 to bring together researchers, educators, practitioners, and more from various disciplines to participate in research, education, and policy. You can find us all over social media if you'd like more information, and please do feel free to reach out. We love expanding our collaborative network. And so the goals for this session are to share our research journey with you all and what we have learned along the way, provide some perspectives, and give you the beginnings of a roadmap to implement this type of perspective in your own research. We'll hear from 3 speakers. And then at the end, we have a panel discussion. So we'll be discussing some relevant issues and thinking about how to appraise any type of microbiome work with this microbes and social equity lens. So our first speaker today is Dr. Monica Trujillo, an associate professor at Queensborough Community College. She'll be presenting some of her work on COVID and wastewater tracking by borough, and she is present in the room today. Our second speaker joining us remotely is Dr. Ari Kozik, a research investigator at the University of Michigan, as well as co-founder and vice president of the Black Microbiologists Association. She'll be presenting on abolishing the use of race as a simple biological variable in human microbiome research. And our third speaker, who is also in the room today, is Dr. Carla Bonilla, an associate professor who just moved her lab to the University of San Diego. Congratulations, Carla. She'll be presenting on culturally relevant microbiology education, social justice-centered science pedagogy using microbiomes. So again, thank you for attending this session, and do please catch up with us online. We're on various social medias. You can reach us by email. And then we also have additional sessions that we're putting on this summer to plug a conference at another conference. And then before I turn it over to our speakers, I wanted to do a few acknowledgements. So the University of Maine land acknowledgement is here, but I'd also like to recognize that we're all coming from universities that are on lands that weren't once our own. So the University of Maine recognizes that it's located on Marsh Island in the homeland of the Penobscot Nation, where issues of water and territorial rights and encroachment upon sacred sites are ongoing. Penobscot homeland is connected to the other Wabanaki tribal nations, the Passamaquoddy, Maliseet, and Micmac through kinship alliances and diplomacy. The university also recognizes that the Penobscot Nation and other Wabanaki tribal nations are distinct, sovereign, legal, and political entities with their own powers of self-governance and self-determination. And then I also wanted to give a labor acknowledgement that I've borrowed from Dr. Tara T.J. Stewart. We must acknowledge that much of what we know of this country today, including its culture, economic growth, and development throughout history and across time, has been made possible by the labor of enslaved Africans and their ascendants who suffered the horror of transatlantic trafficking of their people, chattel slavery, and Jim Crow. We are indebted to their labor and their sacrifice, and we must acknowledge the tremors of that violence throughout the generations and the resulting impact that can still be felt and witnessed today. And so with that, I would love to turn it over to Monica to share her talk today.
Hi, everybody. Thank you so much for being here. So what I'm going to tell you is the work that we started doing in New York at the beginning of the pandemic. And I think that the reason that got us working on this was, if you guys remember, New York was the epicenter. And there was very clearly certain neighborhoods that were suffering the brunt of the epidemic at the very beginning. And it was this desire to contribute to understanding what was happening that was the guiding force for this project. So I am a professor at a community college, and I certainly did not have the resources to do this research, but I really thought that looking at wastewater could be a way to understand what was happening. So I am a good friend of the guy that is right below. His name is John Dennehy, and he's a professor at Queens College. He's a virologist, and I reached out to him and said, do you think we can work on this together? And through John, we also work with Davida Smith, who is here today in the audience. And we started trying to think how we could contribute to a problem that was just at that time having such a heavy impact in our city. So at the time, what was known was that SARS-CoV-2 could be detected in wastewater. We knew that its persistence was inversely correlated with temperature. There was this idea that what it was shed in feces was not infectious, and that was important for safety issues. And we knew that it was stable, even though that's something that we learned later. And what was behind our thought, it's something that most likely you guys have heard before, but if you have not, is this idea that wastewater gives you opportunities to understand about the health of the population that is contributing to wastewater. And I want you to think of this not only in the context of the United States or big cities where there are wastewater treatment plants, but I want you to think about this in places where there's no wastewater treatment plants. But what we do know for a fact is that when you look at a population that is served by any sewer system, what you have is a mixture of people who are infected and those who are not. And some of them can be symptomatic, some can be asymptomatic. Some of them can have the resources to reach out and be treated, and some of them do not have the resources. So the very interesting thing that wastewater surveillance provides you with is a way to look at everybody, right, who is there and to get their characteristics. So what we know is that this, as a disclaimer, this slide is written for SARS-CoV-2, but this is true for many other infectious agents, influenza, polio, antibiotic resistant markers. You know, there are many, many things that can be found in wastewater. So the challenges that you have when you want to use wastewater surveillance is that you are looking at a diluted situation. So you need to think about how you are going to concentrate, how you're going to purify, and then how you are going to measure effectively the concentration, and how you're going to use that data to go back and have an opportunity to become an actionable item when you are looking into public health. So all of this was happening in New York City. And for those who don't know, New York City is a city with significant inequalities, right? And if you look at it, even though it looks like by 2019, things were looking a little bit better, right? There's certainly inequality. And I don't have the data to show you what happened after COVID, but every single thing that we know points toward inequality having substantially increased again. So our work has been done in part in collaboration with the Department of Health and Mental Health in New York City. And this is what they have shown. This is the data, right, that I was just making reference to. Everything shows that not only the inequities that exist in New York have persisted during the pandemic, but the hardship here, what it's listed is financial, right? But you know that if you have financial problems, you're going to have emotional problems, you are going to have family problems, you name it, right? And obviously that impact was distributed differently among different races, among different income sections. And so, yes, there is a recognition that we need to work on health equity, but there's so much work to do, right? There are some papers that are coming out trying to look at measure the impact of COVID-19. And I chose this one because it's probably the black and white, right? If you are dead, there's nothing that you can do about it. And you see that the impact on the life expectancy before and after COVID has made differences that already existed, right? Significantly worse. In particular, I am Latina. I am from Latin America, and I've been living in New York for the last 20 years. And it's quite clear what it meant for our community. So I don't know if you guys remember that during the pandemic, there was initially this talk about how the immigrant communities were contributing to the spread of the disease, right? And the data now show that not only we were responsible of keeping the city alive, but the essential workers were the ones who bear the brunt of the sickness and the disease. So as an example of how unequal New York is, this is today, right? And this is the vaccination by zip code, right? So it is, how many cities do we have in New York City, right? And to be honest, it has to be said that the access to vaccine has been done as wide and extensive and easy as possible, right? But the differences that already existed in the city before the pandemic that have been exacerbated by the pandemic are again reflected on this. So this slides are just to show you what I believe that our research in wastewater, it's so important and meaningful, because if you look at the drainage area, the wastewater treatment plants for New York City, there are 14 of them, right, distributed across the 5 boroughs. And each one has a different, they serve different number of individuals, but it covers everybody. So initially, what we wanted to do was to make sure that we were looking at surveillance. Can we find out which boroughs, which wastewater treatment plants have higher numbers, and therefore can we try to get more resources to them? After that, as the pandemic progressed, we wanted to be sure that we could learn more about the evolution of the virus, and that's going to be the second part of my talk. So when we started, the Department of Environmental Protection in the city had no way of measuring the SARS-CoV-2 concentration in wastewater. And what we did as an academic group was to provide the training for the Department of Environmental Protection about how to do it. I like to mention this because it was a unique opportunity as a researcher to work on something that was making a difference in the quality of the people in the city that I was living, right? I laugh and I said, you know, I've been a scientist for many years, but it took, 60, all right, to be able to do something where I could feel that I was directly contributing to something. So we developed a protocol that was adopted by the city, in which we tried to make it safe and cheap, right? And so we found out that if you pasteurize the wastewater sample, the signal would actually be better. So that solved an important problem, that is how to deal, wastewater is certainly contains a lot of pathogenic organisms. Even if you are not concerned about SARS-CoV-2, there are many others. So that was a big and important thing. This work was also done by undergrads that, had been trained before the pandemic. And because of that, they were allowed to work during the pandemic. Right? So our protocol is published in case you guys are interested. And we did our best to try to find something that was easy to do, but also cheap. So our results are summarized in this slide, where you see that you collect wastewater, you find the virus, you concentrate it, and then you isolate the RNA using exactly the same approach that is used to diagnose by PCR the infection with SARS-CoV-2. You come up with a way to calculate the concentration of SARS-CoV-2 per liter of wastewater. And what you see in the bottom is a graph that correlates the clinical data that the city was getting from the testing with the data that we had in wastewater. But the problem, after the worst was over and we learned that the virus was evolving, was how are we going to monitor the evolution of this virus? And again, in a city like New York City, you can think that depending on where you are, and depending on the job that you were doing, you will have the possibility to work remotely. You will have the possibility of, being tested if you were asymptomatic, or you will not have the possibility of working remote. And if you were asymptomatic, you will never be known because you would be out and about and working and perhaps even spreading. So we thought that what was done by a lot of labs, and it's obviously extremely important, that it was to monitor the evolution, and these are the reasons why we needed to do it, was done with clinical data, right? So we all know that vaccination have an impact on the evolution of the virus. We know that evolution is impacted by the evasion of the antibody-mediated immunity. All these things we knew, right? But the question was, how are we going to study the evolution in a way that we really catch everybody? And the clinical sampling sequencing depends on whose samples do you have, right? And how well they represent the population. And obviously, it mostly depends on people who have symptoms, but even more important, it's relatively or a lot or very expensive. Interesting enough, New York City received significant funding to do testing, but it changed as the pandemic evolved, right? So as you all know, the availability of antigen tests that you can do at home have significantly reduced the number of PCR testing that is happening. So taking all of that into account, but remember that this is early. We started doing this in January of 2021. We decided that based on what we had learned, we thought that we could sequence the signal of SARS-CoV-2 in wastewater and try to study its evolution. So this is what we wanted to do. Our effort was concentrated into sequencing wastewater samples so we could identify the different lineages. And I want you to think about two things. We wanted to be able to identify the variants that we knew were circulating, and we wanted to be able to identify the new variants, right? There is some work that has been done that shows that many times you start detecting the signal in wastewater before there's a significant number of cases that allows you to get somebody into a clinic where that person is tested. So wastewater has some capacity to predict. So what I want to show to you, and this is an idea to try to share with you what you find in wastewater. So each one of these blue things is the virus, and each one of the colors are the spike proteins. And think that each different color is representing a different receptor binding domain. But when you collect wastewater, what you have is the virus that has shed from multiple individuals. So what you have is a mixture. And what we wanted was to be able to do something like this, right? Be able to put all the ones that have the same spike protein in a different group and then quantify. So how do we do this? And the way it's done is after you isolate the RNA, you make cDNA and then you sequence. When you sequence, you have two opportunities. You can do whole genome. That's what is done for every clinical sample. Or you can choose a small region of the genome of the virus and concentrate into that. Besides the differences that you can see here, there's something very important that you need to understand. That is that when you do this type of sequencing, you fragment your nucleic acid. So the big question is how you are going to put it back together. If you are thinking of a sample that's coming from a clinical sample is coming from one individual, well, they are all from the same person. But in wastewater, you have this mixture of everything. So this is what you start with, right? A puzzle with million copies. But what you don't want to do is what is shown on the left, on the right, right? That puzzle, it's nothing. It's not real, right? But you could potentially, you could make it. It would be what is shown here on the right, right? Oh, sorry, on the left. A virus that has different spike proteins in one particle. That doesn't exist, right? But if you think about it, how would you do it when you are getting all these pieces from many different people? So this is what we did, right? So we said, let's focus on a small piece of the genome of the virus that we know for a fact that we get the whole sequence from the same nucleic acid, right? So our first effort that was spearheaded by Davida was to work and amplify 300 base pairs, right, from the receptor binding domain. And then a collaborator that we met had a different sequencing machine that allow us to go for a bigger fragment. And just to demonstrate to you how this worked, I can show you what we did on a paper that is shown in the bottom, where we compare the clinical, what the clinical data show and what our analysis show for different months in the beginning of 2021. And what I want you to remember now is that these pictures look quite similar, right? So we have a proportion, it varies a little bit, but we can identify the main lineages that were circulating. So just to show you what piece we are focusing on, that's the receptor binding domain. And if you look at it, the receptor binding domains contains mutations of clinical relevance, right? So I pick the first significant or one of the important lineages that we saw, Delta and then Omicron. Using this approach, we were able to detect Omicron in the city in wastewater on November 21st. That coincide with the time that South Africa officially said that Omicron was at work, was spreading. But now I want you to look at something that for which we still don't have an answer. That is that when we analyze our sequences in wastewater, we found that certain constellation, certain combination of mutations were not present in the clinical data sets. So what we found was that there was some genome that could not be, had not been found still in patients. And so these are some of the mutations that we found. And interesting, each one of them had been predicted to either expand the host, right? For example, some of these mutations allow SARS-CoV-2 to infect rodents when the initial Wuhan variant was not able to infect rodents. We also found that some of the amino acid changes were predicted to evade some antibodies. So we came up with a hypothesis that this was the virus that was evolving under the pressure of antibodies, vaccines, you name it. So our interest was to find out where they were coming from. And then Omicron came, right? Because at the beginning, before we were talking about this cryptic sequences, Omicron had not appeared. And everybody was saying, well, perhaps what you guys are seeing is an artifact. And then Omicron came, and as you can see on the piece of the receptor binding domain that we were sequencing, everything had changed. So if you compare some of the amino acid changes in our cryptic sequences with Omicron, the ones that have the circle are the ones that Omicron shares with them. So so this was all done in New York, but then this work was expanded in different parts of the country. So California, and this is not updated, but California, analysis of wastewater in California show that, yes, there was a group of mutations that were found in wastewater, but not in clinical data. Missouri had something similar. Oklahoma has recently found something similar. Wisconsin has found something similar. And that brings us back to, again, what is the origin of these cryptic sequences? And I would like to finish by saying that there are two big things that we have learned. One thing is that SARS-CoV-2 can replicate in the gut. And that when we are comparing data from clinical data with data from wastewater, we are comparing kind of apples and oranges, right? Because most of what it's found in wastewater, it comes from the virus that replicates in the gut. And most likely the evolutionary pressure there is different. The other thing that has changed is that we know today that individuals who are immunosuppressed keep the virus in their system very long. And during that time, the virus evolves. And many of these immunosuppressed individuals have a capacity to shed the virus in a higher proportion than healthy individuals. So we still don't know where these cryptic sequences are coming from, but we have, we have advanced in terms of trying to understand it. Something else that we did was we tried to explore what else was in wastewater, what other organisms we could detect the genetic signal, but we couldn't find any result that really suggested that the cryptic sequences could be coming from another living organism. So I need to acknowledge everybody who has contributed to this work. So it involved the schools from CUNY, the City University of New York. That's where I work at Queensboro. That's where John works at Queens College. Most of this work has been done at Queens College at John's lab. And all this, Sharon Cannoli is a postdoc at John's lab, but all the rest are graduates or undergraduates who have done this work. And I would like to say that I think with the exception of John Dennehy in that lab, and perhaps, yeah, I think nobody had been born in the United States, right? All of us are immigrants. And then from the new school, that's where Davida was working when she was here. These two undergrads work with her. Now she is at Texas A&M in San Antonio. And then our collaborator, Mark Johnson, and his postdoc have done the work at the University of Missouri. And I would love to, I have to recognize that it was a unique experience to work with city agencies like DEP and the Department of Health and Mental Health in the city. And I want to tell you that Davida will also talk about this work tomorrow at the same time at 1:45. So if you want, I think it'd be great. So thank you very much.
Today, I'm going to talk about a complex but important topic, which is abolishing the use of race as a simple biological variable in human microbiome studies. So my plan is to go over the goals for the talk. Then I'll talk about why this topic is so important. Then I'll introduce some key terms. We'll do a little time traveling. And then I'll talk through some examples that hopefully will leave you with some things to think about that will result in your continued engagement with these issues. So before we dive in, I just want to affirm that you might be challenged by some of the content, and that's okay. We each come to this conversation with a unique perspective informed by various factors in our lived experience, but it's important to communicate in good faith. You may learn some new things, and I encourage you to give yourself time to process the session today. And lastly, I expect that no matter what, we'll be able to maintain an atmosphere of safety, kindness, and mutual respect. So by the end of my talk, it's my hope that you'll be able to understand the origins of racial categorization in the United States and the limitations of the way it's been applied to scientific research. You should also be able to identify the connections between social equity and the microbiome. So as a society, both nationally and globally, we are lacking in health equity. Health equity is an ideal that all people should be able to attain the highest levels of health. That sounds great, right? Why wouldn't we want that? Unfortunately, achieving that ideal requires us to do something that historically humans haven't been very good at, and that's valuing everyone equally and then intervening when evidence and outcomes run counter to that ideal. So communities of color are disproportionately affected by disease. Puerto Ricans are twice as likely to have asthma than white Americans. Black American children are 4 times more likely to die from asthma than white American children. American Indian and Alaska Native and Black women are 2 to 3 times more as likely to die from a pregnancy-related cause than white women. It's important to note that increasing economic advantage does not fix this problem. The pregnancy-related mortality ratio for Black women with at least a college degree is 5 times as high as white women with a similar educational background. And then we often hear and see Asian as a group descriptor, but the United States Asian population represents people with roots to more than 30 countries, each with their own unique history, culture, and language. It's far from homogenous. And we see inequities here too, if we look closely enough. Native Hawaiians are more likely to die from heart disease. South Asians are twice as likely as white persons to have coronary artery disease. Native Hawaiians and other Pacific Islanders also report having poorer quality healthcare than other subpopulations. So how do we approach the problem of racial and ethnic disparities conceptually, and how does it inform our science? These kinds of questions aimed at unraveling the causes of these disproportionate burdens are usually siloed as disparities research. But there's a troubling assumption underneath a lot of the discourse around these issues, that somehow there are inherent biological differences between racial groups that lead to poor health. This is incorrect. Historically, racial and ethnic classifications have been socially constructed based on perceived biological differences and used as justification for systems of social hierarchy and oppression. Race-based biological or purely genetic hypotheses and investigations have failed to explain or eliminate disparities in health outcomes. Conflation of race with biology in the study of health disparities promotes research priorities that focus on identifying individual or group deficiencies while minimizing or ignoring altogether larger systemic, environmental, or structural forces that can influence outcomes. This framing is common in the literature and in the public discourse, and we saw it a lot at the start of the pandemic. It was clear that COVID-19 was killing people of color at higher rates. It's almost a reflex to assume that this is due to some inherent difference in the affected group that makes them more susceptible, and then to organize hypotheses and research questions around those assumptions. And there are studies out there that did just that about COVID-19. But in order to move towards equity in biomedicine and microbial sciences to promote health, we need to radically adjust the assumptions that inform our approaches. So first, I'm going to define some commonly misused terms. When I say things like Black, white, Alaska Native, Asian, or Hispanic, you likely recognize those labels as different races, or perhaps you'd say ethnic groups. These words are commonly used, sometimes interchangeably, to describe groups of people. So let's talk about what they actually mean. Race is a social and political construct that assigns significance and symbolism to different types of human bodies based on observable characteristics. It is not based in biology, nor is it fixed in time or geography. Racism is a little bit more complex, as there are several different definitions that have emerged in the past few years to try to more accurately capture its meaning and impact. But in this talk, I use racism to mean the belief that race is the primary determinant of human traits and capabilities and the system of oppression based on race. Racism has both individual and systemic manifestations. At this point, most people can identify an individual problematic act. Microaggressions are examples of individual and sometimes unintentional racism. Systemic racism, though, is a system that consists of structures, policies, practices, and norms, that structures opportunity and assigns value based on phenotype, or the way people look. Importantly, systemic racism does not require individual malintent. It is a status quo maintained by inaction and apathy rather than just an accumulation of overt acts of racism. Then there's ethnicity. This word is often used interchangeably with race, but it means something completely different. Ethnicity refers to the grouping of characteristics that people may have in common and can include a number of elements, including language, religion, shared regional history, traditions, customs, and culture. And lastly, there's ancestry. This is often used as a proxy for race, but it should not be. Ancestry is simply the subset of paths through the human family tree by which a person has inherited each segment of their DNA from specific ancestors. I'll talk more about that in a moment. As I said earlier, race classifications take differences in appearance and equate them to real, discrete biological differences, but this is not accurate. In fact, racialization is not fixed in time or geography. The racial categories we use today have changed several times, often for sociopolitical reasons. It also isn't fixed in geography. Before 1930, Mexicans were considered to be white. When Irish immigrants first came to the US, they were not considered to be white. The definition of whiteness changes for sociopolitical reasons. At one time, a person was considered Black if they had even one ancestor of African origin ever. This is known as the one-drop rule. In the United States, we have been categorizing racial identity since the very first census in 1790, reflecting the central role of racialization in American history. And many people couldn't control how they were identified. Census takers would just assign races to people based on how they looked when they came to the door. The prevailing thought at the time was that race was a fixed biological characteristic. People could be sorted into different buckets based on appearance, and society was structured around that idea. And that's why race is actually a proxy for racism. Before we dig deeper there, I want to talk about ancestry again for a moment. So this is a plot represents commonly used genetic reference data sets that are often used in human genetic research. So the colored dots represent the genetic background of these reference individuals in this data set. The gray dots represent a real world genetic data set from a New York City biobank consisting of over 30,000 individuals. And so the important part here is to know that real world data do not cluster along the continental reference data. We do not see separate islands of genetic variation according to continent. So ancestry is actually continuous. It's not discrete. There aren't buckets. It's a continuum. And ancestry is also a historical concept. And imposing categories on genetic ancestry fails to adequately capture human genetic diversity and what we know about human demographic history, in which mass migration and constant mixing across groups have been the norm. So the impact of the history leads to different structures of genetic variation in different parts of the world. So such studies illustrate just how inappropriate use of discrete continentally based categories can be, particularly when information framed as genetic ancestry can potentially influence medical care. Ancestry is also a historical concept, and it takes a slice through one part of time in reference to human history. And so continental groupings and the ways that we think about this can quickly become another way of saying race. You may be wondering, what does science have to do with all of this? Why does this matter to us as researchers? Why am I talking about this at ASM? Well, it's because the scientific enterprise has participated in building the racialized systems of oppression that exist globally, but especially in Europe and North America. Science and public understanding of science is central to society. It impacts government, it impacts policy, it impacts culture, the economy, and law. So what we do, how we ask questions, how we communicate our findings, and the way that things are interpreted has ripple effects through society. Racial classifications didn't always exist. Discrimination and stereotypes always existed, but they were usually based on things like country of origin or religion or culture, not so-called scientific distinctions. But in what we now call the United States, during the time of the American Revolution, European Americans adopted a scientific ideology that humans came in distinct types or races. Why would they do this? Well, because they were facing a contradiction. They were about to stake their lives and their livelihoods on this ideal that there was a natural right to freedom. Put another way, that all men are created equal. But even as they did this, their world building included the enslavement, subjugation, and exploitation of Africans and Indigenous Americans. How did they resolve that contradiction that everyone has a natural right to freedom, but yet you're depriving other people of freedom? It's because of science, or so they thought. Their contemporaries believed that there were different types of people, some more superior than others. And so the division of people into groups based on general geographical origins of their ancestors or descriptions of the way they look is the basis of a man-made strategy for making sense of treating some people better than others. In the 1730s, Swedish naturalist Carl Linnaeus devised a system that showed how living things are related to one another. Linnaeus classified humankind as a species within the animal kingdom. He divided the human species into 4 varieties: European, American, Asiatic, and African. In his view, the 4 were more alike than different, but he believed that they were created separately. This belief in completely separate races was a better justification for racist ideas. His classification of different racial groups as different varieties laid the groundwork for others to use science and classification to try and justify discrimination through perceived biological differences. Look at the stereotypes embedded in each of Linnaeus's racial descriptions. For color, we have red, white, yellow, and black, and very problematic descriptions of other characteristics about hair and blue eyes and dark hair, twisting braids, flat nose, swollen lips, and then behavior. So American, which it would be Native American or indigenous, they had unyielding behavior. The Europeans were wise and inventors. The Asians were haughty or greedy, and then the Africans were sluggish and neglectful. He also believed that these different types of people governed themselves by different things, so by customary right or by opinion. So he believed these things about these different groups, and he made these classifications. But these aren't the groups that we use today, those 4. So enter Johann Blumenbach. He was a German anatomist and naturalist. He invented the term Caucasian in 1795 in his book on the natural variety of mankind. He gave 2 reasons for his choice. First, his feelings about the beauty of people from the region, and second, the probability that humans were first created in that area. So Blumenbach divided all humans into 5 groups, defined both by geography and appearance. In his order, they are Caucasian for the light-skinned people of Europe and the nearby parts of Asia and Africa, Mongolian for most other people from Asia, including China and Japan, Ethiopian for the dark-skinned people of Africa, American for most native populations in South America, North and South America, and then Malay for Polynesians and Melanesians of the Pacific and Aboriginal Australians. Blumenbach changed the original racial classification developed by Linnaeus by adding the Malay category. He also changed the principle of human order from being geographical to a hierarchy of value or worth. So Blumenbach based his racial categories on how beautiful he thought each group of people were. Interestingly, Johann was actually an abolitionist and strongly upheld the unity of the human species against the popular view that races were separately created. So he disagreed with Linnaeus in that regard. And he believed that all humans were the same species. He noted that all supposed racial characteristics vary and change. And he also noted there were not clear boundaries between racial groups. And he rejected the idea that Black Africans had unique features that made them either mentally or morally inferior. However, he still advanced a classification system. It was just based on his preferences. So he saw people as deviations from a Caucasian ideal. Then we've got this guy, Samuel Morton. He was a Philadelphia physician, and he was considered a great American scientist. He supported the idea that races were different species through his work measuring human skull volumes. So Morton believed that the larger skull volume meant you had more intelligence. He used this to rank the intelligence of human. His measurements found that modern Caucasian skulls, the term he used for people of European descent, had the highest volume. Next were Chinese, then African, then South Asian, and American Indian. Morton's work was used to directly support the system of American slavery. If Africans were not of the same species of Caucasians, then enslaving them was more easily explained and excused. This belief supported the reasoning of Justice Roger B. Taney in the Dred Scott decision of 1857, which determined that Black individuals did not have the right to American citizenship. Remember what I said about science informing the law? Here's a great example. Morton failed to understand the significance of human variation for several reasons. One was institutional racism, another was his own bigotry, and third was an incorrect scientific assumption that different races originated in different zones of the world. The last person I'm going to talk about is Samuel Cartwright. He was a New Orleans physician and supporter of slavery and a slaveholder himself. In 1851, he published his report on the disease and the physical peculiarities of the Negro race, in which he argued that high rates of physical and mental illnesses afflicting Black people were products of the supposed biological and mental inferiority of the Black race. In this report, Cartwright introduced what he called drapetomania, which was a disease causing slaves to run away. Cartwright viewed drapetomania as a mental illness that could be beaten or tortured out of those who resisted enslavement. He claimed that Black people had unique traits that gave us especially high readiness for servitude. These claims were not scientific, though they were presented as medical fact. Dr. Cartwright's report tried to justify and defend the institution of slavery as natural. This is the guy who supposedly quantified a difference in lung capacity for Black and white people, and it is the basis of race-based corrections and lung function calculations that are still in use today. But these terrible ideologies didn't end with the Emancipation Proclamation. Journalists, teachers, preachers popularized the findings of these men and others like them. Leading publications clearly communicated the view that Caucasians were innately superior and they were responsible for civilization in the world, and that inferior races were destined to be either overwhelmed or disappear. Frederick Douglass, a former slave and the most prominent 19th century abolitionist in the US, argued against the idea that Africans were less human than Europeans. In the immediate aftermath of slavery, everyone from physicians to scholars and politicians sought to explain the supposed high rates of disease, most notably tuberculosis, among Black communities. According to historians, the diseases of Black communities were therefore thought to be the Black body physically breaking down because we could not handle the responsibilities of freedom. The product of these and many other racist ideas was the eugenics movement, a theory that human population could be improved and human evolution could be accelerated by selective breeding. The US eugenics movement spread their message through incredibly effective science communication at state fairs, at educational institutions, and professional conferences across the country. The movement was thriving and funded and championed by many well-known scientists from the 1880s to the 1930s. It influenced domestic and immigration policy, and it harmed many people groups well beyond the 1930s. It's important that you understand that these weren't fringe individuals. These were well-known and respected scientists and intellectuals. There were meetings of scientists and thinkers, much like this one, where eugenics was considered a progressive, mainstream, scientifically sound ideology and was used to inform policy and practice. This discourse harmed not only Black Americans, but many other marginalized groups. The North Carolina Eugenics Board forcibly sterilized over 7,000 people, who were mostly Black and female, from 1929 to 1974. Tens of thousands of Indigenous women were also subjected to sterilization through other programs as recently as the 1970s. The scientific enterprise has been used on both sides of the Atlantic for centuries to justify the racialization and subsequent oppression of many marginalized groups and to impact public opinion. The way we think about race and the assumptions we make about its meaning and impact are passed from one generation to the next. Society was structured around these ideas, and neither science nor scientists are immune. It is therefore imperative that we uproot this legacy from the foundations of science. Even now, though we have made progress, a lot of existing literature perpetuates harmful or determinist frameworks when it comes to race in the context of differences in health, sometimes by design, other times by omission or misinterpretation. Here's a study that looks at over 200 articles examining race or ethnicity and health that were published between 1990 and 1999. These articles were cross-referenced to the grants from the National Cancer Institute at the NIH that funded them. This timeframe is important because when a number of policy changes regarding health and biomedical research went into effect. The NIH Office of Research on Minority Health was created, the Human Genome Project got started, the inclusion mandate for women and minorities in research was added, and the share of federal funding for the NIH increased. This paper sought to determine how biomedical researchers were using race or ethnicity in their studies at the time, what conclusions they made, and the implications for policies that may be made in response to such findings. The author found a number of patterns. First, there was a lack of definitions. It was unclear what the authors meant when they used race or ethnicity as a variable. Second, some studies reported that they adjusted values or measurements for race and didn't explain why. And third, in some cases, researchers continued to suggest biological differences between racial groups as some sort of a priori knowledge, even when their data didn't support it. More specifically, no author explicitly articulated a definition of race. That is, the investigators did not explain if they conceived and utilized race as a biological construct, a socio-political identity, or a social proxy for cultural or behavioral practice. And further, the defining characteristics for ethnicity were inconsistent across studies. Some people simply use place of birth as ethnicity. So problem number one, no explanation of how the participants are being characterized. And number two, when race or ethnicity was used to stratify data, the authors rarely provided explanations for what their results mean. And even when they did, it was vague and lacking in detail. So if we have a critical mass of papers with findings that link race or ethnicity to health outcomes or health risk, and yet no further discussion of how or why that is, or what that means for medicine or healthcare, then the author's work is vulnerable to misinterpretation in an environment where we should be extremely vigilant given our historical record. This is the antithesis of scientific rigor. The harm this perpetuates is systemic. Here's a recent example of how this manifests today. Both anecdotal data and formal study have substantiated the fact that Black Americans are often untreated or undertreated for pain in healthcare settings. This group set out to explore whether this bias was informed by some type of false belief. They asked a group of white laypersons, so non-medical or non-science professionals, and a group of white medical students at various stages of training to indicate their beliefs about a number of false statements that you can see listed here. You can see the kinds of things they asked straight from the mind of people like Samuel Cartwright. They were asked to rate the pain a white person and a Black person would feel across a number of scenarios. So what they found was that laypersons who more strongly endorsed a false belief reported lower pain ratings for a Black versus a white target. They also studied the medical students and residents and found that half also endorsed some type of these beliefs. Those individuals who endorsed the belief who were in healthcare rated the pain of Black patients lower and made less accurate treatment recommendations. So bias continues to shape the way Black patients are perceived and treated. Here's another more recent example of systemic harm. There have long been questions about the ability of pulse oximeters, the clip device that you place on your finger to measure blood oxygen, to perform accurately on darker skin. They were largely tested and validated on people with light skin. A group of my colleagues here at Michigan conducted a multi-center study to assess the phenomenon and its impact. So they paired pulse oximetry measures of oxygen saturation from two different methods. They tested for hypoxemia or low oxygen. They analyzed thousands of these measurement pairs from Black and white patients from over 178 hospitals. Black patients had nearly 3 times the frequency of hypoxemia that was not detected by pulse oximetry as the white patients. But reliance on pulse oximetry to triage patients and adjust supplemental oxygen levels may place Black patients at an increased risk for low oxygen. The variation in risk according to race necessitates the integration of pulse oximetry with other clinical and patient-reported data. We can't just go by these measurements. And so just last week, a follow-up study was published that showed that across multiple hospitals across the country, there was actual delayed treatment for Black and Hispanic patients due to this discrepancy. Why? Because the pulse ox measurements were what was used to decide who would be admitted to the hospital and who was sent home, who was eligible for a ventilator and who was not, and who would receive life-saving drugs in short supply. So COVID-19 care, including distribution of life-saving therapies, was significantly delayed for Black and Hispanic patients due to inaccurate oxygen readings from devices that work poorly on darker-skinned individuals. Remember that higher COVID-19 mortality rates have been seen in communities of color across the United States from the get-go. There were studies that looked for a biological reason for this. One study claimed that Black people had a higher expression of some gene and suggested that that might be why Black people were so affected by COVID. This uses genetic data to reinforce the othering of a non-homogeneous group of people. This is a perfect example of why we have to think critically about explanations that suggest inherent biological differences associated with race as a mechanism for poor outcomes. There is a human cost involved in the way we operationalize race, which is why it is critical for us to get this right. These were examples of systemic racism. In the last 2 years, we've been hearing a lot about systemic racism. There isn't a singular consensus definition, but most references describe it as relating to policies and processes that create racial inequality. We've gotten a little bit more proficient since 2020 in recognizing and calling out individual acts of racism, personal attacks and harassment, hate crimes, things like that. But we're less proficient in the structural elements, understanding the historical roots of the policies and practices, the barriers to support, resources, environmental injustice, as well as the ways that individual acts of racism both support and are supported by structural racism. It's a feedback loop. Race isn't a fixed biologically determined variable. It's a social political construct that can have biological consequences through racism. So we need to be thinking about how we can approach research questions that account for these realities. Further, because racial and ethnic identity is only one of the identities we have, it's important to remember intersectionality. Systemic racism doesn't operate in isolation. Racialized experiences are also gendered and classed experiences. Bringing us back to the microbiome specifically, in recent years, we've made some progress in conceptualizing environmental impacts on our microbiomes. While we've begun to incorporate other domains like the natural ecosystem and the built environment, our consideration of the impact of race has typically been limited to behaviors, body sites, and group level differences in microbial community membership. However, humans are an ecosystem in relationship with other ecosystems. And as we've seen, racism by design alters our experiences of and access to these systems. So in order to move forward and truly understand inequity, to maintain and restore health, we must shift our perspectives to examine the impact of racism in all domains. But as a field, there has been limited engagement with the direct effects of racism on human health and the host microbiome relationship. In a phenomenal review by Travis DeWolf and others, they find that of the human microbiome literature archived in PubMed Central in the last 20 years, there are over 14,000 papers that mention race or ethnicity, yet only 114 of them, so less than 1%, mention racism. And then only 8 of them specifically mention racism in the context of how microbiome research is conducted and reported. And then of those 8, 5 of them were published just in 2020. So in this study, cited over 7,000 times, microbes and their functional capacities for multiple body sites were compared across select racial categories. Notice how the groupings vary in scale. We've got Asian and Black, large-scale groupings, which encompass a huge amount of ethnic and cultural distinctions. Then there's Mexican and Puerto Rican, which are much more defined descriptors and yet in themselves also diverse. And lastly, white, with no explanation of what is meant by white. Are we to understand that white is the default? Is it the baseline comparison that we use to define deviations from the norm? The authors report racial and ethnic background proved to be one of the strongest associations of pathways and microbes with clinical data. They also mention an association between race and vaginal pH. One of the many problems with this kind of framing is that there isn't sufficient follow-up discussion to interpret what these associations may or may not mean. And so we're left with an important question. Are these microbial differences inherent in racial categories? I would argue that their implied answer as written is yes. From this kind of reasoning, though, the studies of microbial communities across populations, especially when pertaining to the human microbiome and health disparities, they're upholding a flawed model where race determines some sort of biological state that then leads to poor outcomes, rather than understanding race as a convenient measurement that should direct us to closer examinations of social, environmental, and structural context. Since race is not a biological reality, especially as it's wielded here, we need to look deeper to understand what this apparent signal means. When we're talking about biology, we must be incredibly careful in how and why these group labels are selected and analyzed. Here's another example. This paper, cited over 3,000 times, established a categorical precedent in vaginal microbiome research, claiming definitively that Black and Hispanic women have different vaginal microbiomes or vaginal microbes than white and Asian women. This study concludes that these differences in microbes may somehow lead to different disease rates, and it also uses ethnicity as an umbrella term to describe what is actually 3 NIH-defined races and 1 ethnic group. They refer to their cohort as both Black and African-American at different times. And race and ethnicity are conflated and overlap, underlining colonial histories of difference. Race is hiding within racialized ethnicity in this paper as operationalized. Seeking a relationship between racial and ethnic background and the vaginal bacterial community composition was foundational to the study. That was an organizing research principle they were asking. But it also had a large impact in the field. This study gave rise to several subsequent studies, all of which took the racial ethnic groups as given and began to place judgments, value judgments on risky behaviors or good and bad vaginal microbiomes. These qualifications of vaginal microbiomes and the people they came from correspond directly to race and ethnicity. These differences are also being used to explain preterm birth rates or worse maternal fetal outcomes, while failing to account for the well-documented evidence that racial discrimination, chronic stress, and other disparities contribute to preterm birth, especially in Black women. This study coins the term ratio-ethnic, but doesn't define it. It also invokes ancestry inappropriately as a proxy for race. To my knowledge, there was no genetic data included to back up the claims of ancestry of these participants. And even if there was, we've already discussed that its use in this way is inappropriate. A related paper to this one states, our previous analysis of non-pregnant women indicated that race has a stronger association with the diversity of the vaginal microbiome than does socioeconomic status. And also, it appears that changes occurring during pregnancy may render the reproductive tracts of women of all racial backgrounds more hospitable to certain taxa and less favorable for taxa associated with BV and dysbiosis. Since the changes are generally toward a microbiome composition resembling that of non-pregnant women of European ancestry, it is not surprising that the shifts are more striking in women of African and Hispanic ancestry. So a couple of things here. This is implying that European ancestry is the standard and African and Hispanic ancestry is an aberration. We must do better. So we know that social and environmental contexts can and do have direct impacts on biology and microbiology, but it's clear that we need to reframe our thinking about this so that we don't reconstruct race as a biological entity all over again. What appears to be microbiota differentiated simply by race is really microbiota affected by what it means to be a Black or Hispanic woman in the United States. In the context of microbiome research, we often see racialized terms that do not explicitly name race and are imprecisely deployed and used as variables. These are called ghost variables. They function as oversimplified proxies for racial or racist structural drivers and their associated factors. It's often coded in words that seem to refer to geographic or cultural identity or ethnicity. These words are often historically loaded. The underlying structures and factors themselves are seldom, if ever, referred to, defined, or measured. So we need to figure out how to name, define, and measure the underlying structures and factors themselves. As I mentioned, the ghost variable or the category or label is just the tip of the iceberg that hides a wealth of information that is often extremely relevant to our stated question. We need to look deeper at the associated factors that underlie the categories and further at the structural drivers that sustain the conditions related to the outcomes of interest. We use these ghost variables, but we don't really understand, consider, or explore their context. Words like developed or underdeveloped, urban or rural, public housing, inner city, industrial, uncontacted, pristine, or contaminated are really lazily coded ways of referring to things like access to clean water and sanitation, pollution, access to green space, population density, exposure to traumatic stress, so that's like war, poverty, violence, racism, discrimination, access to nutritious food, variation in family structures, healthcare quality and access, the overuse or availability of antibiotics, dietary patterns, variation in housing construction, exposure to the outdoors. All of these things directly impact the microbiome. So these are the things that we need to be studying, the underlying factors that are having an impact on the people, places, and the microbiomes that habit both. These are called associative factors. These are things that we aren't trained to study as microbiologists, but these are the things that we need to interrogate. Why? Because both nationally and globally, humans have disparate experiences of each thing on this list. And we have structural drivers to thank for that. These are the large, complex systemic factors and structures that underlie our human societies. They're even harder to measure, discuss, or analyze. And we typically don't think about these in the realm of scientific research, but that has to change because these drivers have been used to try to make sense of the breadth of human diversity. Race was constructed in service to these structural drivers, so it's no mystery that the data appears to validate them. The data confirms that these forces are indeed at work. So race as a ghost variable also extends beyond the human microbiome. In studies of the microbiome of the built environment, spaces are racialized but analyzed without explicit mention of structural racism or race. So studies investigating urban built environments might draw connections based on the microbiota of the environment and higher rates of diseases like asthma, but not talk about the discussions of structural racism and things like the history of segregation or redlining or pollution or enduring environmental injustice that create the spatial divisions in cities and leads to the health crisis in the first place. Secondly, in environmental microbiome studies, sometimes landscapes are described as pristine or undisturbed or untouched, which erases the contribution of indigenous people who have been stewarding those ecosystems and that land since before the colonial project. So race does have an impact, but it is through the embodiment of racism. The concept of embodiment says that humans are both biological organisms and social beings at the same time. And therefore, the eco-social premise is that clues to current and changing population patterns of health, including social disparities in health, are to be found chiefly in the dynamic social, material, and ecological context into which we are born, develop, interact, and endeavor to live meaningful lives. Another element we need to keep in mind is that because race is a socio-political construct, racism is what we're actually studying, and it functions differently in different regions. All of the things we just talked about in a US-based context are linked specifically to the history of the US. In many other national contexts, racial politics and racism work differently than in the US. The history of this country led to specific structures that impact specific groups of people in certain ways. This means that in other places, the health-related inequality and discrimination processes may affect different groups than those that fit neatly into our categories in the US. Our frameworks need to be therefore informed by the histories of the relevant regions that we're studying. This is especially important to Latin America and other highly colonized locations. Microbiome studies on populations in Latin America often create specific narratives about people, lifestyles, and bodies. We commonly see narratives about deprived indigenous bodies in developing countries or traditional lifestyles that become sick and are at risk. These narratives echo the arguments and ideas perpetuated by colonialism in the region. This paper that I've cited here is a great review that delves into this specifically in the context of Latin America. I recommend it. So for microbiome research, we need to adopt a systems level framework for how to develop and approach research questions. This is important for microbial ecology, for environmental microbiology, for infectious disease, for education, and for policy. We need to do more to understand the context of our samples, to communicate clearly the constraints and limitations of our studies. We need to do some deep thinking and discuss the topics, being sure to include members from the communities that are studied. This is one of the reasons why representation is so critical. Who's asking questions? Who gets funding to ask the questions? Who are you collaborating with? What is the mechanism for accountability for how we engage marginalized communities? This perspective from MSc that Sue brought up earlier, also recommend that as well. It's a great way to start thinking about these questions. So we covered a lot of ground today, but here's a take-home message. Legacies of structural and systemic injustice shape our current scientific landscape. Race is a socio-political and cultural construct, not a biological reality, and it is through racism that race has biological consequences. Assumptions of inherent, immutable, or fixed biological or genetic differences between racialized groups can lead scientific and medical professionals to ignore clear structural and social processes that perpetuate inequity in health outcomes. Race is often deployed as a ghost variable in microbiome studies, and we need new frameworks for human microbiome research that integrate associative factors and structural drivers. And I would like to end with some wisdom from Maya Angelou. Do the best you can until you know better, and then when you know better, do better. It is past time for us to do better. Thank you for your attention.
Thanks everyone for holding. I know the questions you have for Ari, or perhaps it motivated you to think about things differently. So how do I start the timer so I don't go over? Oh, okay, well, we'll start it. So I actually feel, so my name is Carla Bonilla, and I recently moved to San Diego, but in the program it says that I was at Gonzaga University. So I have 10 years of teaching biology to undergraduates. That's where my background comes from. I'm a molecular biologist in training. And I feel really fortunate to be able to follow those two talks because I know you'll see the parallels. Like I'm going to try to focus on other stuff because I don't think I need to convince you, or at least you'll be able to process everything from a different perspective. And I wholeheartedly believe many of the things you heard today, and I tried to use that in my pedagogy and my approach to teaching in the classroom. So with that, I actually want to tell you just a little bit about myself in the sense that I approach everything I do from an intentional, critically reflected place, thinking about positionality. So thinking about where I come from, what is my identity, and how that informs everything that I do, kind of what Ari was saying. How does that inform what questions I ask? How do I ask them? What do I think are valid answers? And all those things, because that upbringing of mine, that culture, that experience, my identities, holding many, means that I look at the world in a particular way. So I have a very strong identity as a woman of color. I am Latina. And as a student, I was definitely involved in a lot of activism. And what I mean by that is being civically engaged and seeing a problem and therefore feeling that you have agency to enact a change, right? So being involved in making changes in your community that help your community as a student. So that's how I approach things. As a pedagogue, as a teacher, I started working at Gonzaga, which is a Jesuit institution, so of the Catholic tradition, which has a humanistic approach, and I'll leave it at that, to education in the in the Catholic tradition. So the students that come to Gonzaga, or many of the students that enter your classroom, have something that they have that they want to achieve as their goals. And so as teachers, how are we helping them get to that goal, right? Not who we want them to be, but who they want to be, and how can we help them do that as a scientist, as a doctor, as whatever their goals are. So that humanistic approach means that I can talk to my students about seeing the intersection between social justice and science, specifically microbiology. And I also try to come with a student-centered pedagogical approach, meaning what I just said. How do students learn, right? What are the mechanisms that help them learn, and how do I measure that they are learning? Not that I'm teaching the students, not the topic, right? Something you probably have heard. And so you can see here so here's some of my students attending a SAGNAS conference. So how can I help them fulfill both their personal needs and their professional needs? And then also you may recognize the March for Science. If any of you were involved in that back in the day, the students wanted to be part of that, and so I supported them in doing that. Again, how can I help them see themselves in the science, I think, can be the theme. And I come from a long tradition of very strong mentors, and so I try to approach it that way. So with that in mind and that philosophy, I also joined the MSc group, and we've been thinking about how to put this into a pedagogical framework. Okay, so this is just taken from short excerpts from a bigger framework that we're thinking about, but what are some of the pedagogical tools that we can use if we want to promote this in the next generation? So I'm probably talking to the choir, the choir right now, right? You guys think that this might be important, but there's another generation, or how do we continue to help people see these things if we think they are important, or make the connections for themselves in whatever way that they do it? So some of the pedagogical tools that we think would be useful in applying these transdisciplinary ideas of microbes and social equity are here. And I will focus on a couple of them in the way that I do things. So examine, again, positionality, who you are in your situation, right? What do you bring to the experience, and what will be the impact of your actions, whether intended or unintended, based on the privileges that you hold, based on the marginalized identities that you hold. How will you experience that? So the positionality that you have, using critical reflection to think about that, therefore exploring how your identity influences your research design, exactly what Ari was talking about, right? Who gets to make the decisions, and how do we prioritize that? We do that by deploying critical feminist pedagogies to develop critical consciousness. This is the section that I will be focusing on. And ultimately, we would like to, right, then apply these social justice principles and praxis and actually do them in the research design, whether it's health interventions or environmental health or whatever it is that you do to eventually apply it. So I want to do this in the classroom. But before that, I don't think I need to, again, convince you why this matters, but I do want to, kind of bring it back that this really matters a lot for ASM, right? So if any of you were at the panel on Thursday night on the IDEA committee, they told us about their strategic plan. This is ASM's DEI. They're not called that, they're called IDEA committee, but looking at issues of diversity, equity, and inclusion. They gave us a list of priorities, and the first one actually is raise awareness about the intersection of science and IDE and DEI, right? So this is really important for ASM. So what we're talking about is not just important to me or to you, but to our society. And they devote, and ASM devotes an entire journal to education as a pedagogue. This is the stuff that I'm really excited about, but journal to looking at education research, microbiology. And there's this whole section on inclusive education. So if this is something that you're interested in, there are a lot of articles that cover this topic. So this matters to us. Okay, so my background then coming from, again, I'm trained as a molecular biologist, but I have always been interested in education and pedagogy. I wanted to use and think about what approaches are there already out there that could do this justice-centered science education. And going back in the 60s, this educator Paulo Freire, if any of you are familiar with him, you would know the work, but I will briefly summarize why this matters. So he was studying a literacy program in farmers in Brazil. So he was a Brazilian educator, and went to these groups of people who were farmers and tried to work with them on these literacy programs. But instead of just saying, here's how you learn, literacy, whatever it is, communication, verbal, oral, he worked with them, right, from this perspective of there are groups, which would be the farmers in this case, the oppressed and the oppressor, right? This dynamic that was very popular at the time. If we apply that though to how to learn, the take home really is that he wanted the oppressed group to, or what he saw was important, was for them to be agents of change for themselves, to see and to learn what was important about them and their community, and to use their own knowledge, right? What we talked about too, the knowledge that you bring, and identify the areas that were important to them. He called them narrative themes. So these things that are important to them and how to use literature and excite them about learning because there was an outcome that mattered to them. Okay, so if you're a scientist, what is the outcome that matters to you? How does your identity inform the outcomes that matter to you? That's one way to see that approach. More recently, Daniel Morales Doyle published this paper where he was working with middle school students and trying to use a similar approach of critical pedagogy. He called it social justice center pedagogy. But he basically, the same approach, tried to identify in a young population, in these students, what matters to them, what are the issues that matter in their communities, and then teaching them chemistry, right? So you can talk about covalent bonds, or you can talk about, and that sounds really exciting as a chemistry major, right? But a lot of people don't find that interesting. But what if we use it as a tool to address issues that matter in your community? That is the way that we can approach this, or the way that he approached it. So one last pedagogy topic. I'm particularly interested, again, coming from the experiences that I've had I'm especially interested in culturally relevant pedagogy. So I'm going to call it CRP from now on. That will be the only acronym, or the second acronym I will use. Again, as a molecular biologist, I'm inclined to use those acronyms, but culturally relevant pedagogy was developed by Gloria Lansing-Billings, and she started working on this in the early 90s. And basically her work was looking at this population of middle school teachers that worked with a predominantly Black population of students. And the paradox there was that she was looking at these classrooms and these students who were doing really well, right? They were thriving in Philadelphia. They were doing really well compared to what people thought was the norm. Again, going back to kind of what Ari was saying, these preconceived ideas that we have about biology that are really structural, societal structures. So these teachers were working with their students, and the students were doing, were learning, were achieving to the levels that the school standards, were meant to do, and they were just really thriving. So when looking at these students and the teacher interactions, she saw some things in common, and those are in the yellow over here. And so all of this is summarized in many of her writings. Initially with the Dream Keepers, and then more recently she has put together a collection of those. But originally she identified these three things that the teachers had in common in the way they approach the students. So it wasn't about style, right? It wasn't about how they were engaging necessarily one-on-one, but what they expected out of their students and how they brought out the best out of their students. And so, of course, like any good teacher, she wanted to measure student achievement, that the students were learning, right? We are never gonna let that down. We're gonna hold those standards. The students should be learning something, and so making sure that they do. She also noticed that, Lansing-Billings also noticed that every one of these instances, the teachers had cultural competence, right, with the students. So they understood the students either on a personal level due to shared experiences, or at least were involved in the community that the students were in, so that they could understand where the students were coming from. And also, and this is key as well, and it's part of other culturally inclusive pedagogical strategies is to bring that cultural competence into the classroom and allow that to be valued as part of knowledge in the classroom, right? So just because the students know something in a different way doesn't mean that it's not true, even if it's jarring to us. Trying to really pick at what is it that they're saying and say, oh yeah, you do understand this, you're just using different language, right? And so that cultural competence is something that all of us can do, or we can try to strive for. And then the key to what makes a CRP different than other inclusive pedagogy is critical consciousness. And this is, I skipped that earlier, but that is actually very as a key of what Freire's philosophy is. He called it conscientization. So that's also a term in English, but it is to be critically, so using critical analytical skills to being conscious of the structural systems that you exist in too, okay? So this may sound like a lot to us of the students, but I'm gonna break it down in a little bit so you can see how students, in this case, they were 4th graders to 6th graders, were able to do all these 3 things, and that led them to be really successful students. So if we break down critical consciousness in the classroom, now there is a lot of literature on this, and this is my summation of sort of some of the bigger, broader concepts, but you could break it down into sort of these 3 or 4 concepts. So critical consciousness begins with, first of all, identifying the issue, okay, becoming conscious that there is a problem. If you can't see that there is a problem in society that you're trying to address, then, you know, we can't have that yet. So many people actually, I can see this, right? They read the news, they live the experience, they don't need to read the news, it's happening to them. They know that there is something that is wrong in the system. There's something wrong at school, there's something wrong in the medical system, there's something wrong in their job. So identify that social injustice that is happening. Where this gets, where I want to bring it back to that positionality and critical reflection is that you want to understand why it's happening, right? Because you can see that something wrong is happening, but you don't know why. And you haven't reflected on how your experience allows you to see that other, that's somebody else's experience, right? I know what I go through, but how do I identify with an LGBTQ person's struggle, right? What is it that I bring that either allows me to see their struggle or prevents me from doing so? So having that critical reflection about your own position in order to see the experience. Agency is huge. Being able to realize that you have the capacity to enact change. This is where a lot of young people get stuck. So there's a lot of adult developmental research on this, but you may feel apathy, right? And those are comments that people often make about young people. Oh, they just don't care, or da-da-da. Well, it can be overwhelming to see these huge things happening to society, whether it's war, poverty, whatever it is. And if you don't feel that you have the agency to make any change, you get stuck at that point, and we're not making that progress as human beings. So realizing that you do have the capacity to act and have an impact on your community. This was important for Freire's work, that those farmers needed to feel like they could make a difference in their own community and improve their own situation. Again, being mindful of your intention and impact, because a lot of us have really good intentions, but where we come from, what biases we bring in may have unintended impacts that we did not mean to have. Ultimately, you have to actually take an action. That's this is where there's some debate as well as in critical consciousness. Do you actually do something about that? So becoming active and participating in civic engagement for the betterment of the community. So you may hear that there's something wrong. You may feel that you have the ability to make a change as a scientist, as a researcher, as a policymaker, but then actually taking that next step. It's a lot to ask. And this is a process that happens throughout your life. It's not something you just do one day. Okay, so let's see if we can do any of this with students in the classroom. So these are the students who who joined me in this effort last semester. And these are 10 biology students, junior students in a seminar course that at Gonzaga University. So this is not an introductory, I'm going to give you a little bit of background on the class because it might inform what we were able to achieve. they don't have microbiology background, so I had to bring in a lot of microbiome, but had to bring all of the microbiome background to them. Being at a Jesuit institution, they have a little bit of background on social justice, but now we have to apply it, right? That's the thing about this interdisciplinary approach, is that you want to integrate both things and make them one. And so that can be challenging. And so, yeah, here they are. And this is on the last day of our class. And I'll tell you sort of how I went. So the rest of the talk will be about planning the course. I unfortunately cannot share any of their data with you, but I'll sort of tell you kind of what the lessons learned were learned from that. So the other thing I want to bring up, which again, thinking back to what Ari was saying about your experience, the experience you're having right now, right here, is that we were going to talk about really controversial things that could come up, right? Because they might challenge what the students believe. So Gonzaga University is a predominantly white institution with students of relative means, okay? So sort of middle class students. So here we're going to talk about racism and socioeconomic status and all of that. So how do you engage in those conversations, being respectful, being mindful, and still being willing to learn, right? Because if you are confronted with a challenge, you might shut down immediately. So how do we create that environment? And typically at Gonzaga, because it's a liberal arts school, we often sit in a circle where we can see each other, and we want to engage that dialogue, which is really important. So the physical space matters, how we look at each other, how we engage with each other physically. We were able to be back in class and wearing masks the whole time. In the picture, they just took off the masks for the picture, but they were very glad to be wearing masks. So I had to remind students to be and train them into having productive dialogue. These, again, are fields of their own, but how do you engage in listening and not arguing with people and not shutting down when you're challenged? And so we did some reading on this type of engagement and practiced in having those conversations. They really like talking about that, which was really funny, talking about how we're going to talk. But it meant that they were willing to talk at the end, right? They were able to say the things that they meant, which is a challenge when you are engaging a group in DEI conversations, if any of you have in the past, much less in the scientific realm. And also having different formats of engaging, right? So journal clubs, we did scientific journal clubs because it's a science class, roundtable discussions. We did that more to the second half of the class. Think, pair, shares. Any of you who know what that is would hopefully think that that was a good idea. Otherwise, is where you give people a little bit of time to think about the prompt, right? Because something they need to process. This is, again, we're teaching people. So you need to give them some time to think about the prompt and process it. Then they pair up and then they share. So this is an easier way to engage with someone because you're just telling them the thing that you thought and you're not talking to the whole group immediately. And then we would open up to a bigger group discussion. And this applies to anything you're teaching, math, history, whatever, right? This is not a social science thing. It's just an approach to teaching. Sometimes I actually ask them to write what I call 1 or 2 minute papers, which turn into 5-minute papers because they just get writing and writing and writing. And I didn't want to break them up and say, okay, you've written enough. Now we need to talk, because they were practicing, right, that written communication. So giving them different ways of practicing what they were learning. So I also use backward design to design the class. So not just like, hey, we're going to read a bunch of microbiome papers and let's see what happens, right? Which is kind of how we start teaching. Instead, thinking first, what are my learning objectives for the students? What do I want them to learn? That is the first thing that you do when you're doing backward design in teaching. So you identify the desired results. And I will get into the more detailed ones in a little bit. But basically, the students should be able to problematize the research on microbiome and social justice topics and propose solutions. So I used this term earlier and I didn't define it. What I mean by problematize doesn't mean to just make up something and see it as a problem. Instead, right, is to see a situation and understand the historical context of it, right? Why does it work that way? The stakeholders, who is it impacting and who is involved in this issue, and then understanding where solutions may come from. So again, I think Ari did an amazing job at explaining why race is a problem in microbiome research. So just problematizing the issue, although we probably already thought it was a problem, but understanding where it's coming from and coming up with solutions. So the students should be able to do that at the end of the course. These are brand new students, right, in biology, but no microbiology training. How can I, then the next step in backward design is determining acceptable evidence. Okay, so I need to know how will the students tell me that they achieved the goals that I had for them. So the students can deeply discuss, and I meant deeply, and I'm really a big advocate for that, because oftentimes we have really superficial conversations, right? We don't say what we mean, we kind of use terms and we don't know what they mean. And you're like, okay, what just happened? Which can be sort of a barrier to discussion classes, because you're like, what was that about, right? What did we just talk about? Because people don't really engage with the content, don't use the terminology correctly, or don't really explain what they mean. So the students should be able to deeply discuss and explain the concepts in both written and verbal forms. And then from there, you decide what the class is actually about. Okay, like what content you're going to use, what book you're going to use, what the exams are going to look like, because you have come up with this plan. Okay, so if we break down those learning objectives just a little bit more, because that sounds like a lot. Okay, so this is a process that you go through as a teacher, right? So you describe, I want my students to be able to describe the areas of microbiome research and human health, climate, environmental sciences, using field-specific concepts, right? So they should be knowledgeable of the terms that we use. They should be able to explain the techniques, right? So very, very basic bioinformatics and statistical methods to measure microbiome diversity and composition. Those are the kind of assays they're going to be looking at when reading microbiome papers. How do the researchers do it? How to interpret those PKWs and so on. Importantly, they need to be able to identify the connections between microbiome research areas and social justice concepts and the reciprocal interactions between them. They should be able to propose new research areas and interventions. And they should be able to critically reflect on their own learning objectives. So that critical reflection was where it was a little harder for me to get them to do, but at least I could get them to reflect on their own learning goals. So to see how that influenced their growth as students. And they should be able to communicate these things, obviously. And so again, you would use all these to help you design your class. Okay. So I'll quickly go through sort of the curriculum topics we covered. And I won't get into what microbiomes are and all that. A lot of you know what those are and get in here, but I do want to point out that we basically covered a lot of topics. They really were students were really interested in nutrition and diet and the effect on health. That class was very interested in human health. They were interested in stress and the effect of stress. So the gut microbiome, the brain gut microbiome axis, they really like that, and mental health connections, the effect of reproduction, so reproduction and microbiome, that was something that they were very interested in as well. Antibiotic use and microbiomes was something. So these were the kinds of topics, the kinds of papers the students read. Primary literature is what they used to understand these ideas. I also had to then integrate ideas or introduce to them at a much deeper level. So it's not introduced, but actually get into more deeper level social justice principles. So I had to introduce those topics to them, and we had readings. So all of this means readings, right? I had a reading for them, and then they would either answer questions or have a discussion about it or write a paper. And the 5 principles of social justice that we use were equity, access, participation, human rights, and diversity. And so there is quite a bit of literature on this. If you or yourself are not aware of, how do you define social justice and what does this mean, right? I would suggest that we start from a grounded place, and then now we know what people mean when they say social justice. But understanding, that human rights, this participation one is actually important because you may think that someone has access, you may think that a group is diverse, that has people from many different identities, but are they really able to participate, right? So these are actually different things that all feed into social justice. And we also use the One Health framework to try to tie these things for them, to try to see the connections between environmental health, human health, and the way that structural structures that prevent human health are also involved. So sort of some examples of learning activities and reflections that the students engaged in. You can imagine this was a lot of reading on my part and grading, right? But basically the students, in order to demonstrate understanding and application of the concepts, they would lead a journal club, just like we've been sitting listening to really beautiful science. The students should be able to define and describe and explain what data meant and use scientific rigor to do that. That was more of a summative assignment. They also had to lead groups, group discussions, social justice topics. This was a little harder because they are not trained at that. They are biologists, not sociologists, right? So this was a learning curve for all of us. And that was more of a formative assignment. Can they do it? Can they get experience at doing it? To demonstrate content knowledge, they actually graded that they were writing properly things and that they got a lot of practice at that. So a lot of assignments where completion with lots of feedback was all that they would get. And then at the end, they wrote a paper that was in the format of a perspective so they could apply these ideas and show me that they had learned how to talk like a scientist. And then the critical reflection that they engaged in was really more metacognitive, to learn about the process that they themselves went through, because I want them to be life learners, right? So what did you do? How did you come into the class? What skills did you gain at the end? And they get points for this, or not a lot of points, but I do want them to basically also at the end, they had to explain to me how their perspective had changed through the course. Some of them took the course because it's what fit into their schedule and it was there. They were honest about that. But at the end, did your perspective change a little bit? So, like I said, I don't have any data to show you yet, but sort of some of the things that I saw coming out of the class. Assessing critical consciousness development, right? This is a process. This is something that you develop as a human being. Many of the students identified a personal connection, and I learned this in their writing because they told me a lot of what they were going through. They identified something that mattered to them in high school, in their family history, and so they had that connection that mattered to them. Henry, the person on the top left there, he shared some of those things with me in writing. They were motivated to act in civic engagement. Some of them actually ended up volunteering for things because they felt that this was something that mattered and they could take an action. So remember, that's the second step of critical consciousness, to actually, well, the last one, to engage, but to feel that you have the ability to make a change. And then they also applied prior academic experiences to the class. They would often come in and talk about their sociology class, their criminology class, and apply those concepts into the discussion. So it made for a really rich conversation because they were already comfortable with all the terminology from the social sciences, and they started talking about it in this class, and it was really great. And with that, I would like to acknowledge all the students in the class who were very willing to engage in the challenging discussion with me and each other. Jasmine Newsom was my undergraduate research assistant, whom I had many conversations with about how to do this in the classroom. She gave me her student perspective. And then also other folks, members of the MSc group, whom we've had conversations with talking about this pedagogical framework, and Mindy Howard at Gonzaga University for discussions about biology education research. And so thank you for your attention. And I think we'll take questions now.
Carla, if there are no questions in the room, perhaps okay. We also had one that came up on the chat. So when we get a chance, just let us know, and Ari can read out the question and the answer that she typed to that.
Yes.
Thanks. Go ahead.
Thank you. The question is for Dr. Trujillo. I was wondering if there have been any evaluations of the impact, especially differential impact, of showing the different rates of SARS-CoV-2 in the wastewater. I know that those sewer shed areas do not coincide necessarily with differences in socioeconomic status, but can you elaborate on that?
Yeah, so it's a very interesting question, because
now the data is available publicly, but there was a lot of pushback from the city agencies in terms of identifying the sewer shed. So if you look at my presentation, I never show anything that allow you to know in which sewer shed we saw this cryptic sequence or this other one. So as always, when you try to address social issues, first of all, I think it's important that it's not up to any scientist to share that data without taking into account the context. But on the other hand, there are other interests that are at play. So it's an excellent question because it brings right to the surface one of the problems that we have. As a scientist, I did my best to find out what the truth was. As a citizen, I need to agree with the city agency that I'm working with in how to share the results. I don't know if I answered, but
So we have
if
a
when you ask the question, you could identify yourself, I think it'd be great.
Hi, my name is Emily. I'm from Penn State. This question is specifically for Dr. Kozik. I hope I'm pronouncing that correctly. Thanks everybody for great talks also. I know there's a huge need in clinical research to study non-white, non-male people. And contextualizing in your talk, I'm wondering if you have any like ideas or resources for how to go about making sure that we can have some kind of accountability in clinical research, like when we're designing a trial, without kind of, I was thinking about your categorization of race, and that's how we normally kind of get medical demographic data from what's in medical records, things like that. Do you have any kind of suggestions for how to move forward in that way?
Yeah, so that's a really good question. And I think that there's two challenges, I guess, stratified in time. The first challenge is that because of the way we have done science and leaned so heavily on these categories, there's not a lot of extraneous data about prior data sets that exist that we base like new studies on. So like we can't go back and like figure out other like context about older studies. But what we can do is kind of like start now as we design new studies. So I think that, you know, asking or collecting data about racial identity is okay and important because, as I said, when you collect that, that allows you to study racism because you can study someone's experience as a racialized person in the context of whatever clinical thing that is that you're interested in. But it's just important that you don't say that their background is the explanation for whatever the outcome is, especially if you don't have good information about their specific context. So like when you have people come in for a study, knowing, you know, where they live, like what are they exposed to environmentally? Like if you have a Black population, like what people who would I self-identify as Black come from a very heterogeneous like group of people. That could mean, you know, someone like me who's been in the US for a few generations since enslavement, or it could be like an immigrant whose history here is more recent. And so, but in terms of clinical data, Black is all we get. So if Black is all we get, then we lose a lot of dimensions of data that can be important. So I think thinking about in terms of your research question or what it is that you're trying to study, what other dimensions of data would be important or informative or useful, knowing ahead of time that just relying on the racial categories will not be sufficient to help you answer your question. So I think thinking about it that way can help in study design. And I think, I mean, hopefully one day we'll have a lot better best practices around how to do this. And there'll be some accountability on, you know, the IRB side, the funding side, the institution, the research support side to get there. But that's where we're going to need to go.
So we were wondering, we could take the question from the
Q&A that you guys have?
Oh, yeah. So it was similar actually. So it says, as someone who has worked on a vaginal microbiome project, how can I aid in making sure that collaborators and scientists are analyzing the microbiome from an unbiased lens when the baseline, in quotes, of the research is from the viewpoint of previous studies using a white vaginal microbiome as the standard? And so my response to that was that because the previous studies used data from one group, and like I said to the other question, we can't go back and create variables from data we don't have. Then the introductions and the discussions of papers surrounding this old data need to specifically state and articulate that this particular group that's being studied is not a standard, and to be sure to clearly define what the study question is and whether there are unstated assumptions that may be inaccurate that are underlying the way that you think about the implications or the conclusions. And so then that also includes giving suggestions for future work that include some of the other associative factors that I talked about that may impact the microbiome, even if the participants in the study are white. As I mentioned in the talk, like white and whiteness means different things in different contexts, depending on what year you ask the question, like what is a white person? So like thinking specifically about what does that mean? Like what is the actual background? What is their social and environmental context? Those kind of descriptors to give the population more context would be helpful. And then new studies need to be designed to think about this from the get-go. And then the other piece of that is I think we're going to need to, as a field, come together to think about methodology. Like how do we do analysis with such diverse populations? Like a lot of what we do is based on reducing the dimensions of data, not expanding the dimensions. So like statistically, how do we do that? And I think looking at trying to borrow from social science or anthropology, where they have been thinking about how to talk about people and how to measure things about people for longer than we have, I think will be a good way for us to start figuring out how to change our standards for the way we talk about things and study things.
We have another question in the room.
Thank you for excellent talks. I've been following the work on the distance during the pandemic. So many important issues. Actually, following up a little bit on what you said, my question is if we can develop guidelines, some more ethical or lists that it's difficult to do checkoffs, right? But if you do qPCR, there is a QA/QC protocol. Is there certain ways that we can help integrate what we have been talking about instead of just trying to understand, but do a little more and try to push the field? I think there are parallels in all three talks on this. Thank you.
Carla, do you want to answer that question?
I'll add just a little bit. I think something that I didn't do, it was underlined, is ethics training, right? Like really, what does that mean? We're not training that. Yeah, ethics isn't like, don't mess with your data, right? Like we're taught that, don't fabricate data. But how to apply it to human studies or anything like that? We're not trained like that in undergrad. And so at a curricular level, that's important. And thinking back to the different committees that ASM has, maybe that's something that we really need to think about in advocating for, in the curriculum for microbiology, right? Because ASM has a lot of things that they have to say about microbiology education. That would be one thing, but just as a undergrad level. Yeah. Anybody else?
I think that for everybody, for the younger generation, I think being, strong enough to use terms like social equity instead of social equality, and being able to explain why you think that is important in your conversations with your peers, with your mentors, and afterwards when you become a professor. I think in that I agree with Ari, in that it is the new studies, the ones who can start to show the way. There's no doubt that it's going to be difficult, right? But I think that just choosing the right terms, addressing the real problems, even if we are limited in our solutions, it's a step in the right direction. Ari, do you want to add something?
Yeah, I would just add that I think thinking about the stakeholders in the scientific enterprise and how much influence they have and what can they do in their domain of influence. So, I mean, like funders is an obvious one. Like what kinds of studies get funded? Like what is considered to be innovative or important or relevant? What are the significance or the broader impacts of like work, valuing this kind of work and this kind of scholarship specifically in the scientific space is really important. It is difficult, often I find, to convince some folks that these topics are relevant to microbiology. Like I have a mentee who was told that like, you're here to study microbiology, you're not here to study social equity. So there's a bifurcation of like how these things move together, how they are important and they are linked. And so I think the funders and the institutions, they are the ones that set the priorities for what gets studied. So on their level, that can be a thing. Publishers, they are the gatekeepers to scientific communication. I talked about earlier how scientific communication is critically important because that's how science learns about science, but it's also how the public learns about science. And we can do a lot of narrative shifting based on how things are covered and how things are talked about. And so what can publications do to advance the way we talk about things? What is the minimum acceptable amount of discussion or qualification that needs to happen when you're describing differences in different groups of people? That's something that they can do. PIs, a lot of training of trainees, Carla said, this isn't something that's taught often in part of curriculum. Can you integrate this kind of training into your lab or into your the way that you do things? So there's something that everybody can do depending on whatever level of power you reside at. And if we all push together, we can move things forward instead of depending on the new generation of trainees to fight through all these layers to move things themselves. Like everybody's got to be pushing together. And so I think that it is beyond time for us to really make some decisions on how we're going to go about doing that.
We have one more question and we have reached the limit. I'm not kicking anyone out, but if you have to go somewhere, don't feel like do what you need to do. But yeah, one question here.
Thank you so much. I was wondering, I thought it was so interesting, your slide talking about how there was no actual regional basis for a lot of these determinations of race and ancestry. I want to be able to keep talking about it after the conference. And one question I had lingering that I want to be able to respond to if somebody asked me about it, if with those genetic testing sites like 23andMe, et cetera, ancestry.com, where then they do try to pinpoint where these are from. And so I was wondering what weird or incorrect or false, et cetera, statements are they assuming, and how can I respond to them if I get that question later?
Okay, so there is, everyone get your pencils or phones, I guess. It's 2022. There is a review that was published in Science that is called, where is it? It's called Getting Genetic Ancestry Right for Science and Society by Anna Lewis. It was published not a few months ago. It is a really excellent introduction into this issue. Ancestry is really complex because of all of the things that go along with it and genetic risk and like diseases and things. And so they explain it much better than I had to read the paper several times to be able to distill the information. But basically, I think it's related to how like ancestry is a continuum, but it's also a historical concept. So someone's ancestry includes every ancestor they've ever had, And so there's a lot of it's a challenge to like tell someone, okay, and pinpoint here, this is the place where your ancestry originated, when really it can go back so much farther than that. And so the companies like that, when we think about, what is my genetic background, like that is taking a slice out of human history and focusing just on that slice and like the mixture at that point in time. And that's not necessarily correct. And it also assumes that there are pure gene pools associated with certain continents of people. And so now we'll have terms like, oh, well, there's people that are admixed, like there are people who have African and European ancestry, like me. Like I am African American, but the person who owned my family was from Ireland. So I also have European ancestry. So that is considered admixed. But is there a like Irish gene pool or a European gene pool that is free from mixing? No. So thinking about what it means when we use those words is really important. So the review again is Getting Genetic Ancestry Right for Science and Society. Everybody should read it. It's really great. And I think that you will find a very detailed way to argue this point with people when they inevitably bring up questions. But that's a great question.
All right. So I think that we used the time that we had. We appreciate a lot your participation. We just, I think I'm talking for the four of us. We just hope that this would be the beginning of a conversation because it's quite clear that there's a lot to do. And that I think that each one of us in our own field finds we get challenged by the things that we say after we take the time to think about. I think that each one of us, or at least I'm talking about myself, that I realize that there are things that I haven't gone deep enough to really be contributing to create a more social, equitable world. So I think it's great that you guys took time to be with us today. Thank you very much.